Anybody who knows like,
Ma'am, alpha helix, beta sheets,
then
turns loops.
loops. Then other than that, like do you guys know any like any secondary structures? So these both are the major secondary structures, right? But in helix itself, we are having return helix, pi helix, like that. Two different extra additional thing we are having. So only these are like helix, turns, loops, and sheets. Helix, turns, loops, and sheets and turns are there, right? These are the only secondary structures we are like going to see about now. So now in a sequence, if they are giving a raw sequence or you are identifying a sequence by yourself. So how will you identify your structures? Like we don't know anything about that sequence. We just want to know like study about that sequence, like how it is looking and what are the properties it is having. So see, if you want to see, then we are interested in the structure also in the sense. Then first we will try to go with the secondary structure, like whether it is predicting and in like by the algorithm whatever it is predicting and the real life situation is right. That will be like having too much of variations. That also I have to say because based on some concepts or like some algorithm that it is predicting that structural thing. But if you model the structure or if you try to generate it in using some other tool, then that will differ. So we have to think about that. But for basic analysis, we can do the secondary structures. Okay, how the secondary structures there? Like we will see about Chow-Fasman algorithm and the Gore method for predicting secondary structure and Cybrid. Three tools we will use to predict those secondary structures. Like all the tools are predicting the secondary structures only. We will see the difference. So every also have some different things, right? So that's why we are going for three tools. Like basically, we will go with Chow-Fasman, Gore, and neural network is there. So neural network and Gore is almost similar. So we are neglecting that neural network and we'll go for that. The Gore method, and another tool that is there called Cybrid, which is predicting the secondary structure based on like that is also like the algorithm is based on neural network. So the software one is based on statistical method. So remember that the statistical method we are taking is like Chow-Fasman. Using the tool Chow-Fasman, we are statistically identifying those things. So that is by Chow-Fasman. And using neural network, we are trying to use predict by like Cybrid. Cybrid we will use, right? That will be based on that algorithm which is behind the tool is based on neural networks. And Gore is also predicting based on like statistical method. Okay, Chow-Fasman and Gore are both are in the same method of identifying the secondary structures. Like, have you studied about propensities in your class? Like, and did they taught you about propensities?
Yes, ma'am, a table kind of thing means
Yes.
we identify with the
Yes.
ratio and all we do.
Yes, yes. That is like we will actually you performing the thing is very simple. Like it's a chop person method and the core method as well as the separate. Just need to give input as a sequence, then it will give you what are the helix are there and what are the sheets are there. So what is that B address? Okay, while performing one thing you will see, okay? Let me try to present that screen. Okay, now we will see about chop person method of predicting secondary structures. Yes. This tool is a CFSSP, Chow-Fasman Secondary Structure Prediction Server. Okay, this server we are going to use to predict our secondary structure of the protein. Okay, so simply I will give one sequence and let's see what kind of results it is predicting and how they are calculating based on propensities. I will tell you. Okay. Um, you want to check for any sequence in your wish? Like you have any idea to check for the protein?
Ma'am,
Yes. How do
uh, ma'am, can we go for the RV3371 protein?
that is like structure we are having, right? For that protein.
Yes, ma'am.
Then no need a prediction, right?
Yeah.
Like we already done the crystallization structure means they have already predicted the whole thing. If we don't have any some like it's fine. We can check for that also. What is that name? RF33. But the
No,
result
ma'am.
won't be the
No,
same.
ma'am. RV3371. Let me type in the. That
Okay.
one.
RB337. Can you say the name of the protein? It'll be better for me.
RV3371 and RV0183. The both are mycobacterium protein.
Okay, you're working under Krishna Sala.
No, ma'am. I am not working under it.
Okay, okay. Okay. Diacylglycerol acetyl transferase, that protein, right?
Yes, ma'am.
Okay. Okay. Can you able to see the results? Yes.
Yes, ma'am.
Like if you see, we have given the input sequence as this one, like from M to H we have given. So now what this tool has been predicted. The query is the thing we have given, and which will be the helix region, which will be the sheet region. E is represented as sheets, okay, beta sheet like sheets, and T as loops, turns, and C as coils, whichever is not like predicting, then those things will be coils, and some things like will come in the turns, like those are turns. And if you see here, like why they are giving helix, and E, and T, then finally they are giving the structure as helix. This is how they are calculating the propensities. So the how the exact like the algorithm works in the sense like I will try to explain you. Try to I don't know how far you can understand. Let me try to like to my possibility. If you're not understanding, just tell me. I will stop and again I will tell you. Okay. So what it will do, for example, first think about we are having this stretch of sequence up to L we are having. So generally this like Chauffeurin method, how it will predict helix. First I will tell about helix, how it will like judge means. It will choose 4 to 6 contiguous, which means 1, 2, 3, 4 amino acid or like up to 6 it will choose. So after choosing this 6, it will try to create propensities for this thing. Which in the sense, the P of H. P of H we will say it as propensity of helix. If it is greater than 100, if these four the residues, after calculating its propensities, if its value is lying like more like above 100, then this region will be put into an helix region. Okay. Likewise, it will check for sheets also, and whichever is having maximum value, like if P of helix is high in this region, like the propensity of helix is high in this region, that will be the final result. Likewise, it will calculate for like 4 to 6 contiguences, like continuously. First 4 to 6, then next 4 to 6, like that. Likewise, it will calculate first 4 to 6, then next 4 to 6, then next 4 to 6. Likewise, it will calculate. Or else, just put it in a simple words, how we will calculate the propensity in the sense, if we are having 20,000, no, not 20,000, 2,000 residues of, like our protein sequence is around 2,000. Imagine we are having 2,000 sequence. In that, 500 alanines we are having. Okay, remember that we are having 4,000, no, like the whole sequence is 20, like in that one we are having 1,000 helix region we are having. Okay, 1,000 helix region we are having, and in that one itself 500 alanine residues we are having. So for that 500 alanines, it will calculate the propensities. So whether which will this alanine region will go in helix region or in sheet region. Likewise, only it will calculate. Can you able to like understand the thing? I don't know how far you are understanding. Like because by saying, I don't think it's possible for you to like imagine those things. Is it like clear? It's just an algorithm, right? Like if you like sir have it like did he taught us regarding the second estimation?
Yes, ma'am.
Yes,
Ma'am,
sir.
I have one question.
Yes, yes.
Ma'am, in the helix, in the first one, there is a gap between the helix. So is it like if it does not exceed 100, so it will not be a helix, but it can be
Yes.
like sheet, beta sheet or something, right? But
Something, yeah. Like if you see in this region, like we are showing here like gap is there. This is what like Anna was talking about. Why they are having this space in between. Right, that's what your question, right?
yes, ma'am.
So, like these amino acids will are there, right? It will also go to the propensity same as like helix. P of S is equal to P of S is greater than 100 or not. But if it is falling like less than 100, then it will not be coming under helix region. Then the same way it will check for sheet region also. Whether this, like here if you see the like gap is more. So from L to this G region, like the propensity of making like falling of that residue into a sheet region is very less. That's why it is like making as a gap region, and it is checking for turns also, whether it is possible to be placed in turns. If it is possible to be placed in turns, only three residues from this region. Like in this gap, we are having two regions, right? Like two amino acids. For these two amino acids, the possibility is there to fall on turns region. So then it will come in a turn. So anything is not like above it is like empty and empty, and at the T like only two positions are there. So which is having by comparing all these three, we are only having T. That's why this like this amino acids will be put in the T region. Okay, can you able to understand this thing? And which is not whichever is not falling under that region will be like found as a coil. Is it clear?
Yes, sir. Got it.
Okay. Like anybody is having any other kind of doubt regarding this thing? This is how the algorithm works. Like because now you can submit the sequence and you can tell them like total number of residues we are having. This 316 residues are there, and like in helix region, total is 2446, and we have getting 70% of helix and 60% of beta sheets, and turns 10% of residues are getting in turns. We can say it to them, but while understand like working that is like main thing. If you understanding, then that's well and good. Is it clear, right?
Yes, ma'am.
Yes. So some residues, if you can you able to see, like they have mentioned in star region. Can you able to see some G, some H, and some V? Those residues are there, right? So the possibility of these residues are there, right? They are like high, which means they come like definitely these regions will fall under like wherever they are mentioning, right? Like propensity, like whichever it is having like star, then like it means that it will definitely fall under that region only. It won't go back to like it won't fluctuate in that region helix or in the other like turns or coils. It will directly like it will definitely fall under that sheet region. So if you see this V, correspondingly it is making with the E, like beta sheet is there, right? So this V will definitely will fall under the beta sheet. That is what the stars are mentioning. Okay, this is the scale of that thing. Okay, tell
Ma'am,
me. Yes, yes.
ma'am, in this one, so how for presenting that it can be a helix, we are taking six amino acids per time. So, but how about when it's a single amino acid? Is it like some
No,
other?
no. From this six only it will calculate. So if you consider this six, and from this six it will calculate, right? One by one only it will calculate, right? Even if it takes six continuous residues also, like one by one it will calculate. In that meantime, like the G might be having the higher propensity comparing to the other residues.
Okay, ma'am. Got it.
In that way, like it will fall, like make that residue into falling under that region. See, everybody is like falling under that region, but there is like the highest amount of value will give you the exact result, right? Like that, it will predict the same thing. Like the formula you have no right. P of P alpha, like P, and in suffix we will write alpha, and like propensity I is equal to P of alpha by amino acid A of AI by P of alpha. Like that formula is there, na? Like the if you see the definition, the tendency of an amino acid to be in helix or beta sheet, that is propensity. Okay, the tendency of an any amino acid to be in helix region or in the beta sheet, that will like lead to an that is propensity. Is it clear this way?
Yes, ma'am.
Yes, ma'am.
Yes. This is how like the algorithm works, right? So it will have like if you see the browser matrix and all you're creating, right? Like it will start with the initiation and propagation and back like tracing. We will say, na? Yeah, back tracing. First steps we will use to calculate, right? Like similar way of time, it will go for that also. Like same thing it will do. And but for calculating the beta strand also, it will follow the same thing. But in some algorithms they will fix for beta strand to like here they for helix they put 4 to 6, right? As a contiguous residues they will have, right? In some conditions they beta strand they used to put 3 out of 5. Like the contiguous will be 3 to 5. Like that they will put. and the value for V of H is like more than 100, right? Like they're calculating in 100 terms. But for stand, they used to calculate this V of E is greater than 1, like 1. If it is more than 1, it will be coming into the sheet region. Like that, the algorithm behind them, like if you see the values of those things, then you can able to see. Like you will understand by reading itself. Just trying to make assign like understand that whether it is possible to be in this region or that region. This is how you have to perform for your studies, okay? How to predict the secondary structure. And you can ask the question whether this is like this tool is like highly accurate or not. We don't know, right? Because it's just a prediction tool. But this top postman is having some 50 to 60% of accuracy only. It's not having the whole 100% of accuracy. Okay?
Ma'am,
Yes.
uh, ma'am, whatever protein we have to give in this tool, that should be experimentally validated, or we can give the putative also.
No, it has
Like,
to be experimentally validated. Just we are predicting here, okay? It's because we don't know anything about that sequence, that's why we are predicting. So
so
this
we are,
like yes.
ma'am, we are predicting the characteristic of that protein, right, ma'am?
Yes.
Okay.
So if you see your whatever your protein you are having, for example, if you are having crystal structure for that one, if you compare those two results, it might vary because whenever you are going for like a real like experimental thing, that will definitely vary from whatever you have predicted. Okay, so that's why the accuracy of this tool is like from 50 to 60%, not 100%. They're like in that tool, so they're saying they're having only 50 to 60% of accuracy. Sequence only they're calculating. But the nature of the thing, they don't know, right? Like they can't predict the nature of the like each amino acid. Each amino acid in different conditions it will exist in different properties. Like if they are living inside, like for example, think about if a protein is inside the cell, it will have a different property, and if it is outside the cell, it will have a different property. Like the hydrophobicity itself will change, right? If it is inside the cell, then the hydrophobicity will be less. If it is outside the cell, then the hydrophobicity will be more. Like that changes will be there, right? In environment, like think about the real environment, then that will differ, right? For each and every environment, the nature of the protein also will adapt to that one. Then only it will not degrade. So here and now they're not thinking about those degradation and all. They're just predicting this will fall under this region, this will fall under this region. All the bioinformatics itself is a prediction, right? Anyway, we have to validate these results too experimentally. If we are proving only, so that will be the accurate results. Is it clear?
Yes, ma'am, completely clear.
Okay. Then the same way will work for like the Gore method also. So Gore, who his name was why we are saying this is Gore in the sense like his name was Garnier and Robson method. Like he only identified this method. That's why they have given his name for this thing. Okay, let's check for that one. Gore method. Gore secondary structure prediction method. Secondary structure by Gore. Okay. This tool CIBCS. Okay. 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 Is there, right? This is this company is like handling this course secondary prediction method. So the sequence we have given here, right, for the guy he said, right, we can give the same sequence here, but the format has to be different. Like here we can take the foster format, but for here we can't take the foster format. You just have to paste your sequence. Okay. We'll try to submit and see like what kind of results it is showing. Then we can. Okay. See if you see, I have already told you, right? We are having some different helices like 310 helix, pi helix, beta bridge, extended strand. Those things are like this is E represents the strand. It doesn't matter. So here they have given you specifically in that page. So in this page, they're only discussing about helix, strands, and turns, and coils. But if you see in this page, they're discussing about 310 helix also, pi helix also. So what is 310 helix and pi helix? Like if we are having continuously three helixes together, like if you're having three heads together, then that will form a 310 helix.
Ma'am,
Yes, yes.
could you please share this link in the chat box, ma'am? I am not able to open it in my laptop,
Oh,
computer.
okay. Because sometimes like are you facing that
Yes,
site
ma'am.
not found?
Yes, ma'am.
Okay, okay, okay. Wait. Um, because for me also, like previously I was studying, also facing the same problem.
Got it, ma'am.
Yes. Mm, like coils are there, right? The first thing has been predicted as coils here. But if you see here, this thing has been predicted as helix here. Can you able to see the difference? Like in that Chauffeur's it predicted here as a like helix region. The same thing we are predicting, but with the different tools. So each different tool will give us different results. The same thing here has been predicted as a coil. Can you able to see the difference for each tool it varies? And here this alpha helix is predicted as 36%, but in the older results, that alpha helix is presented as like 70% of like residues of falling under this alpha helix. So here, which means like each algorithm is following a different pattern to predict those results. So what about this God prediction? Like whether it is, we can see this is also having not having that much high accuracy. This is having some 60 to like 80% of accuracy it is having. It is also not having the exact accuracy rate, but the score method is having only from 60 to 80 or 75, like that kind of accuracy only it is having. And it is also following a different kind of algorithm, no, statistical method only following with a different algorithm it is like trying to, like they will name it as like so I told you right why they're mentioning 310 helix and pi helix like that. So these things will be included in that algorithm. That's why the results is varying all the times. And they, if you see here, random coil is 222, which means like random coils are high. Then your extended strand and your alpha helix. So after generating, this is only for secondary structure. So after structure, tertiary structure is different, right? Like if you compare with secondary structure, tertiary structure is totally different from the secondary structure. We try to put that in Ramchandran plot, or like that is a validation tool. We used to say like we used to check whether our predicted structure is right or not. Those things we will see, right? Those things will be we can check through a Ramchandran plot. That will predict another kind of that will give you the results. Like, but the accuracy of the Ramchandran plot is a bit more high. So after predicting your structure, like whatever structure you are predicting, you have tried to validate the structure through like a Ramchandran plot. The accuracy rate for that one is very high because it is calculating based on their like backbone of each amino acids. We are having like the arrangement of amino acids, you know, like the peptide bonds we used to say, right? NH, that thing is there, right? That backbone amino acids. I don't know how to say it. What you will say for backbone? Wait a second. But I'm not getting the word. Once I'll get, then I will tell you.
Ma'am, peptide residues.
Yeah, peptide bonds. Yeah, peptide bonds will keep on residues. Those backbone residues they will calculate based on that backbone residues. So that accuracy level is a bit higher than these two tools. This is only to predict the secondary structures. So we can do the same thing for Cypred also, and we can see what Cypred is predicting. And this Cypred used to work based on neural network, another different tool, like different algorithm it tries to follow. Why it is not coming? SOAPMA, everything is like second step prediction rules. We can check like one by one also we can see, but doing the same thing again and again is like the waste of time. That's why I'm not giving you that SOAPMA and PhD. PhD is also working like it has been integrated with GOR. Okay, this PhD secondly. So this is another neural network thing. PhD. We can do the second step prediction here also, but it's a GOR method because those Pravi is like handling all those tools. Can you see the Pravi ID? Like those things are handled by those same company. They were handling those things, so almost the results are like similar. That's why I'm trying to check for cycle. Cycle is not working. In cyber, like service is not clear, I guess.
Ma'am, there is a bad refresh.
Yes, I'm also facing the same problem. I also couldn't get the site. Okay.
Might be they have some server issue.
Yeah, cyber issues is there, I guess. That's why it is not opening. But in Cypher, we will have other extra things also. Like Cypher is like a bit advanced, we can say. They are having some columns we can check for like cellular localization. Also, we can check whether this like amino acid will like lie in the extracellular region or intercellular region. Those things also we can check in Cypher. Right? If you see the Cypher web page in the sense like the like now I couldn't able to open like maybe in later classes we will see like how to like in
Ma'am,
meanwhile
ma'am,
also
in images you can show
yes I can
example.
show right yes thank you for the idea yes it is right अरे यार see the image is not clear this will be the like web page of that one I don't know how far you can see if you see here we are having some things right like domain prediction this MEM sat this is like membrane helix prediction this we will give you can see if we give this one if you click this one then while giving the result it will show you like whether it will lie in like in the extracellular region or intercellular region the amino acid which amino acid lying between like extracellular region and which amino acid like holding inside the residues because all the amino acids like we can't say all the proteins either it will lie in the inside or it will lie outside membrane proteins are there right some part will be inside and some part will be outside in between it will be stuck in between the membranes right so like TLR4 you have you like listened anywhere like about 4 or any other protein some what kind of protein we can say interferons also we can say for protein some region will be inside the membrane region some region will be inside the cell some region will be outside the cell so those residues like how many residues are lying in those regions also we can check through this right uh, so the results will be somewhat like this only the picture is clear I'll show that one oh god it's very bad we will do again this experiment try to do that one again and then we can see this kind of results also it will produce Like if you see here, the configuration, like if it is presenting in the helix or which region, like sheet or like beta sheet or something, this will be predicted like this. This kind of image also it will produce. Nowadays it is producing this kind of image. The tool has been updated, so now it has been producing this kind of image. Whichever region is lying in the helix region, that has been highlighted as pink, and sheet region has been highlighted as yellow, and the other regions are the disordered regions or coiled regions, we can say empty. Like this kind of results also it is producing. Once again, we will see this tool and we will check like whether we are able to get similar kind of results or more accurate results than these two tools like Chow Fastman and Gore. Okay, that one we can see again. Mm, so these three tools we will like discussing about. With Hyper we didn't work now, so we'll make a we'll do the class like whenever we are getting the server like able to access the server, we'll do that one again. Then what else we will see today? Like the secondary sexual prediction is okay, right? Like can you guys able to understand this, right?
Yes, ma'am.
Yes, and you can do in your free time. You can just check. You can take one crystal structure of like whichever we have the structure we will take and take that sequence and try to give it in that tools and try to predict whether it is predicting accurately or not. You can check that way also whether they are giving you the proper results or not. So, but you will find the difference. Like you'll definitely find the result because accuracy is like less when we compare to the experimental structures. Experimental structures are the exact one, but these things are not that exact. Just we can learn. Like these are methods are there to learn those things like raw sequence because everybody used to develop something from the raw one, right? So by that time we can have this kind of things, and sometimes you can predict properly also. Like whatever you're getting results in like Chow Passion method, it may be applicably correct in some experimental side also. Can change that in the research. Anything can change, right? So that will happen. Okay, then shall we see about binding site prediction, binding site identification? Can we see that thing?
Yes, ma'am.
Yes. Okay. So what is this binding site identification? Do you guys have any idea about binding sites, active sites, binding energy? Have you studied those things in your classes? Maybe you might have studied those things. Like MD simulation you have done, like the session.
Yes, ma'am.
Then you should definitely know about this tool, CASTP, right? Docking you guys have studied, right?
Yes,
Docking.
ma'am.
Then you should know about this CASTP also.
Yes, ma'am.
Again, then you have to explain. Who is gonna ready to explain that to me? CASTP. I'm ready to hear. Who's willing to? You had last semester or this semester? CASTP, like CASTP, like MD simulation, docking and all. This semester you studied or last semester?
Ma'am, we studied like we were taught this a few weeks ago.
Few weeks ago, no? Then you can tell. No problem.
Ma'am, it shows the binding pocket, ma'am, of
Hmm.
ligand.
Okay, of ligand. Okay,
Uh,
so what is the use of that?
no, sorry, ma'am.
No, no
It
problem.
shows,
You can tell. No problem. Whatever you are thinking, you can tell.
ma'am, it shows the how our ligand is interacting with the protein in a pocket form, ma'am.
Yes.
With
And
the this server especially predict the uh, topological, sorry, this surface protein.
okay. Then any other answer you are having? Like other people. Pablo said his answer.
Ma'am, it
Any
is
other thing?
based on the volume occupancy actually
Volume
of the protein.
occupancy. Yes. Then any
Yes,
other
based on
answer?
that, the
Tell,
pockets are
tell.
created actually.
Okay. And any other people? From girls, nobody is talking. Why? You guys don't want to talk to me, huh? Nobody is speaking. It's okay. They're not ready to talk with me, so that's fine. Okay, then we will. Okay, they're normally having 2 PCK. Default, they are having 2 PCK. We'll go with this. No, we'll go with some other thing. 440. We'll not find 440.
Ma'am, before going to this, ma'am, you haven't saw the any other neural network based protein structure prediction, ma'am?
Oh, PhD you want to see?
Yes, ma'am. Can we go at least one?
Yes, yeah, we can go, no problem.
Yes, ma'am.
Because the Cypred is working on less neural network. That's what I was about to. Cypred algorithm is there, right?
Yes, ma'am. But the server is not
Okay, not there. Okay,
one.
we'll go with the PhD then.
Yes, ma'am. Okay. Ma'am, but experiment we can mention about Cypher. Okay, if it's work, then we can analysis by our own and we can put the result.
Yes, yes, sure.
Okay, thank you.
Because I have told you this PhD secondary structure prediction method is also like it has
Ma'am,
been.
I think it's working now. Like it's opened for me just now.
Oh,
Cited.
who? Okay,
Can you
Nafis,
share the links or no?
let's share it. Share it
Yes,
and I will
ma'am.
also try to open.
Yes, ma'am. It's working. The same link only.
Okay, then I will also try. Yes. Okay, can you able to see the Cypher page? Yeah, I'm also getting. So if you see here, whatever we are clicking, sequence data we are giving or PDF structure data we are giving, that's up to our wish. So if we go for PDF structure, those are like solid structures, then we can find out like protein metal ion contact prediction and membrane protein orientation prediction and protein hotspot prediction and custom generated file. So for now, we are going to give you only the sequence data because we are not considering about the tertiary structure and quaternary structure. We are only thinking about the secondary structure. That's why we are going with the sequence data and input. If you see here, popular analysis, what they have given you. For this sequence data, we can predict the secondary structure. Pivot 4 can predict secondary structure and disordered prediction like the isoprep. Disordered region, if it is there, it will predict that region also. And MAMESAT is the membrane helix prediction. And PGen Threader is profile-based fold recognition. What is this profile-based fold recognition in the sense? Like we have you studied about patterns, right? In your classes, we'll have certain kind of patterns. If we are having 10 sequence from different organisms, like imagine we are having 10 sequences of keratin from different organisms. Starts from human and its engine, some musculus or something. So for 10 sequences we are having and we are aligning those 10 sequences and we are trying to create a pattern or profile for that one, which means have you seen that profile thing, like let me type it here. uh, so how the profile will look, actually the pattern or profile will look in the sense, if you are having 10 sequences in the sense, is it visible? Okay, so
Yes, ma'am.
yes, I'm putting V here and I'm filling here with uh, SA, sorry, Kalpita, SA. Again, I'm putting here X. Again, I'm putting one hyphen and then I'm putting again on X. Again, I'm putting with the flower braces. SD. So, for example, you remember this as a like profile, which means from 10 sequences, we are trying to create one profile. So this bracket V in the sense, this is in this place only V will come. And this place SAM will come, like from all the sense sequences we are having SAM, and at the X position any amino acid can come, like either T will come or A will come, anything will come in the X region. And ST in the sense, for the following two amino acids S and T should not be there, it has been neglected, like from the thing. Without S and T we can get we can have any kind of amino acid in that position. Like have you seen this thing kind of thing in your classes? Things I have already taught you, I guess, regarding this profile and patterns.
Yes,
You
ma'am.
can say
Yes,
patterns,
ma'am.
yeah. So this in this tool, like where is that tool? Wait once again. I will go to that page. So this pigeon threader profile-based fold recognition. In some cases, we might for our sequence, we can read about the folds and all, right? If it is having similar kind of fold, like it will predict like that. So that is why we can check for pigeon threader and all. But this is like around tertiary and quaternary structure. There only we will see about the domains and all, right? But now we can go for, like, we will check for this memset also, whether it is lying in, like, how the things it will look. And input data will give the same sequence what we have paged so far. Uh, okay, we can test the FASTA sequence here. Then we'll try to 16th November, due to some computer migration, server is down for 48 hours. Okay, can you guys able to submit that thing? I couldn't able to submit.
No, ma'am, the same error.
Same error you guys are facing, right?
Yes, ma'am.
Yes,
Okay,
ma'am. Server is completely
down,
down, ma'am.
down.
Yes, ma'am.
Okay, so okay, then we can shift it to some other thing and see.
Yeah, but we can try by our own.
Yeah, you can try by your own and then we can. So meanwhile, I will try
Yes,
to
ma'am.
do this PhD. Like what kind of result it is giving, then we can move on. Okay. This is because alphabetical things. In this tool, only the amino acid should be there. No need of any faster forward. So we got the results for this thing, and we are having the results for all the pages also. I will share one by one. And here, what kind of results they are predicted. If you see that here also the result varies, like like drastically we can say. The beta strand prediction here, it is very less. It's around 10% only, but in that GOR method we can see that as like 13%, and random coil it is 192, like 43 days of amino acid is there, but there it was random coil was like a bit high. 222 residues were there. Okay, let me show that page also. Yeah, this page. This is click core method of prediction. 22, like 49% of amino acids is lying in the random coil region. 164 amino acids are lying in the alpha helix region. But if you see in this page, you see 206 residues are lying in the alpha helix region, and here it has been reduced. This is how this algorithm works. So every now you can see that every algorithm is having a different way of identifying their things. And one more thing you can see here is extend the strand. No, same thing they are giving you. And also they didn't predict any 310 helix or pi helix. I think in SOAPMA we can see, I guess. One second. I will check it in another server. Take quite time, I guess. Instead of doing this PhD, you can go for that Cypred and see like what kind of result that is giving in neural networking. Okay. And also, we're not facing like all the three tools have same thing, right? So it is the only thing then predicted is like beta turns. It has given you some residues are falling under beta turns. Some in between residues, some 21 residues, it is falling in that. But all the other things are like almost similar. It's a like a bit variation from the. This is a SOAPMA server. Again, the structure prediction. This is also another tool that has been like taken care of by this Brapi. It's fine, like no problem worrying
Ma'am,
about
I
it.
have shared one link. Uh,
Yes,
this NNVPVD.
and then BPD.
Yes, ma'am. NNVPDB.
Okay, okay. Let me check that one. Is it okay, neural network based protein prediction? The PDB validation also they are giving. It has been raised from SRM. It has been created by some student, I guess. Okay, let's check it.
From SRM University.
SRM University.
Yes, ma'am.
Okay, let's try. We don't want to waste your time also. We'll just try. What kind of results they are giving, we can check that also. Before their project they have created maybe. With the prediction and the addition thing they added here is like a validation they are giving. Let's see what kind of validation they are giving and see. But the accuracy rate we can't tell them. They might have taken the accuracy rate from another server or I don't know. Let's check it. I'm also new to this page. And in your whole research or any kind of thing, it will play only not it will not more than play 1.5 percentage of time for predicting these things. Okay, so don't worry much about the servers and all. In your free time, you just read about the treated helix, pi helix, and turns. Those things you just read about. Have a glance of what of those things are there. You can have a look up that one. Instead of telling always helix and sheet, you can tell this kind of turns is also there. This kind of treated helix and pi helix is also there. And some special secondary structures is also there. No? Those things also you can tell them. It's taking quite. We'll get one. We'll go for the other thing and then we'll see whether this is going okay. Okay. We were in class 3. Atlas offers topology of proteins. You know we can do for our sequence also. uh, uploading our PDB file also we can check, not sequence, like if you have model structure you can upload for that one also you can check, and we can directly take it from PDB also if you have that ID, we can directly take it from ID. Um, it's very big protein. I'll go with some small protein. It will be easy. Is it fine for you to explain? Okay. Actually, this is this is a kind of tubulin protein. Here they have shown you the whole structure. That's why it is showing like this. All the chains have been like visible, like in visibility. That's why. But think about this is the only chain. Okay. Oh, it's not clear. Okay, let me go with some other thing. Okay, now the pocket has been created for a chain B. This is alpha tubulin. This is a tubulin protein, 1JFF. We see it is from defined structure of alpha beta tubulin, zinc film sheet stabilized with Taxol. So here if you see like the area they have given and volume they are giving. So what is this like red color balls represents this in? This will call it as a cavities. You can say it in another name called cavities, which means like empty spaces. So in this way only, like the space is showing, right? In this way only, your ligand will travel to bind with your protein. So if you're giving a ligand, it will not directly go and it will not. It should not clash any other things, right? Because whenever a thing is moving, it should find its own path to reach its destiny, right? It should not like make like accidents in between. We can say it in that way. It should not disturb your helix or sheet. Like if for example, think about if I want to do make like if I want to reach here, we just think in this way. So I want to reach this region. It's not high picking. Imagine I want to like go to this region, but I should go without disturbing anything. So for that, I need a way, right, to come to this region. So that region we'll call it as a cavities. That is called what we will call as a pockets. Now, can you able to understand like what is pockets? Like whenever your ligand is coming through, it should find its own path to come and reach its destination. Like in between, if you're having 500 amino acids, the binding site will be like 2-3 regions only, or it might be from like 10 to 20 amino acids only, like the binding regions. So for reaching that region, it should not disturb all the other amino acids because all are molecules and we'll say all molecules, right? Everything has its own electrons outside their like energy. Every element is having in like electrons at their energy like outer valence shells, and that might interact with other things, right? So that's why it's better for to find its own way without disturbing anything. You can go and bind to that region. That's how our like system is also like performing. If any like foreign particle is like affecting our body, it is not like going to like disturb the whole system. It is only targeting a particular thing only, then only it can go and like infect that system alone. For a simple cold flu or any virus, it cannot affect the whole body, right? Maybe we will be having like cold, but you won't be having a body pain like the whole day or something like that. Only like a part of thing only will be affected. So that's how like the nature is like working. It should not disturb the things to reach its destiny, right? This is how the like amino acids also like the ligands also will go and bind to their original destiny region without disturbing the system of protein. That is what we will call it as a cavity. So how this is going and binding? Like if you click show pockets here, it will show you how many pockets are there here. So we are having around more than like 174. 174 pockets it is showing. But why we are only choosing this pocket one also we can say. Like if we can click pocket two and give update, then you can see one more circle, like empty space here. Can you able to see? But in pocket one, if I want to check the like amino acid which is present in the pocket sequence ID, you can see here like the whole pocket A is having this number of amino acids, which means the pocket one will be the like highest amount of highest amount of interacting residues will be there, because the cavity will be a bit more wherever it is finding. This is a taxol binding region. That is why it is showing at the end. If it is in between somewhere, it will make a find a way to go there also, and it won't disturb the other like wherever it can only disturb like it can only touch the amino acids of valine 23, 26 of asparagine, and glutamic acid like 27, the glutamine, and cysteine, leucine from 217. It started from 23, but suddenly it has been jumping to 213 and 217, and it's continuing so on. Then it is reaching at the final 284, and at the final it is reaching some 300 and sorry. This is to second chain has been started. Don't know second chain. Tell.
Ma'am, whatever you are showing, that is the inbuilt protein, na? I mean,
Yes, yes.
okay.
That is a crystal structure protein.
No, ma'am. This protein name already provided on the database, or you are using your own protein?
No, no, it has been provided in the database. I have
Okay.
using PDB ID, right?
Yeah.
So that has been like it has been crystallized structure. So this is accurate
Okay.
results only. Like what? Like it has been already predicted. This one. If you want to check for your like, if we don't know the binding region and all, right? By the time only we will go for this region. Like I'm just telling you to show like how the pocket is working and how it is going and binding this region. That's why I'm telling you exactly this is a taxol binding region, because I already know that this protein is having the ability to bind with taxol. Like and the residues it is showing, right? Those also belong to that region. That's why I can tell you like already it's a known structure.
Okay, ma'am. Okay.
For without like if you don't know the structure in the sense, you can model the structure and upload it in that show pockets. No, no, show pockets. It's in another tab is there, right, for calculation. Okay, let me show the tab. In calculation, the region is there, right? To click here, here will show upload a molecular structure in standard PDB format, which means like the 3D structure. Now multiple NMR structures, please. Non-polar hydrogen atoms will be ignored. We upload your file and check. Okay, and the values will be like from 0.0 to 10 will be accepted. This probe radius, this thing you can check it for your own protein. Okay. And here, okay, that thing has been changed to 1CYSS. So this is only the chain B I'm showing at the end. You can see the residues what are all involved in this like cavity region. Starts from 23 and it will ends in some 372. Only these residues are in the interacting sites. So it can travel in this way. That's why while doing the docking and all, why we are choosing this tool, it's not only this Caspi is there. Some other tools is also there like MetaPocket or something is there. We can use those Meta Finder. Many tools are there to find these binding sites. We can choose that one also. It's better if the structure is already like has been studied and something. It's better to go with the literature interaction sites, because that has been already proven now, so that will be like more accurate. And you can see here the red highlighted regions are there, right? Those are like traveling in this region, so it might touch these regions. The like while the ligand has been traveling, right? It can cross these regions, and suddenly these regions will not be involved because the path of this region might be like a bit distant from this thing. As I, again, it is facing these amino acids. Again, facing it is these amino acids. This kind of things are there. So this will be like it's a tertiary structure. I can able to see like it's like maybe the region which is not showing it. Maybe the 20 beginning amino acid like that. It will be there. So while you're giving your docking file or somewhere, you will give that in to find out the binding site. You will just give this in the tool and take the residues from that region and give it as a binding site region. Okay, is it clear this tool?
Yes, ma'am.
Yes. Hmm. this is binding site identification using passkey. And what else we have to see? This is how many screens are done? Okay, nine, right? Nine. 9 or 8. The second such application is a complete one experiment. Okay, ABC, you put ABC, in A put chocolate, in B put God method, and C put cycle. The next is binary prediction, binary identification. You just put binary identification and use this Caspi as another experiment. I will give you the IDs. What are the things you have to search? Well, okay. If you have to, you want to go for same protein for all the three tools for predicting second structure, or you want different IDs, whichever you want.
Ma'am, whatever you said, I have used my own protein ID.
Okay, if you want to use that one, you can give, or else okay, I can give you one more idea. Also. Whoever wants to use that idea, also you can. If you got the result for that one, it's fine. You can have this result and you can check it.
Ma'am, is it necessary to put your protein ID in experiment, or we can use our own?
You can use your own also, but to be in uniformity, I try to give that results. If so, you can use that same thing. Like how many of you performed the same thing, or only one person performed today?
Ma'am, you can give one ID for the three tools. We can compare, right?
Yeah, you can compare and check.
Yes, ma'am. That would be great.
Okay. I will give you one ID. You can probably it's up to you. If you want it to be in a uniformity, you can make like name that one, or else that's up to you. Okay.
Okay, ma'am. Okay. Actually, ma'am, I have used different ID for the different web server.
Why is it so? Like for all the experiments
Ma'am,
previously, for today only, no?
yes, today.
Okay, no problem. To maintain the because now it's a record kind of thing, right? So they will ask you the uniform results. That's why I suggest you to go with the same thing. It's not a big deal. You can use that thing also. But to be on safer side, it will go with the same thing. Sometimes they might ask, sometimes they won't ask. Use this for second sector prediction, okay? This ID.
Okay, ma'am.
Then for Caspi, putting the video. Use this for Caspi, okay? Predict the packets, binding sites. So it's also already a solid structure. But this also all details are there. Try this one.
Ma'am.
Yes.
So, secondary structure prediction is exercise 8,
Yes.
and binding site identification, that is exercise 9.
9, yeah.
Okay.
You put ABC like subtopics ABC as like for chocolate, no? God and the cypher, okay? Like let's see in next class we'll be able to access cypher or not. So if you're getting results, it's fine. You just try to place those results, okay?
Okay, ma'am.
Yes. Then for next next we will okay. We have finished up to 9 experiments, right? Binding site identification. The next experiment we are having about uh, sequence patterns and profiles, generation of sequence profiles, as well as we are having a few only left out, I guess. Let's check it. And what about like, did they say you anything regarding like this has like practical session also will be shared by any other faculty? Did they give you any information regarding that thing?
Sorry, ma'am, can you repeat it?
Like while taking this BSc paper now for the people, like who are all shared the paper? Syed sir as well as PTV ma'am, I guess, right?
No, ma'am. Only sales, sir.
Okay, only Syed sir took the whole session of sequence analysis.
Yes, ma'am.
Okay, then I will cover those things. One second, let me check your syllabus.
No, ma'am, actually, the part of it was taken by PTV ma'am at the end,
At the
like unit
end,
one, some parts of unit one. Theory.
okay, part of unit one, right?
Yes, ma'am, and rest of the part was like taught by Said sir.
Okay, once again, let me check that thing. Okay, ma'am took only that first like experiment, like first unit, right? Like we brought some parts. Databases ma'am took, or I don't know. It's okay. You don't remember. You finished your exam, right?
Ma'am, yes, ma'am. Database
Then
part
it's
only,
fine.
ma'am.
Okay, no problem. Okay, now we'll finished up to 5 in the syllabus. It shows like 5. And we have generation of sequence profiles using SciBlast, FlyBlast, CanProcyte, and PRAT. Then protein motif and domain analysis. That is there. Up to some F experiment is there. A to F. MEM, emotif, interpros, CanProcyte, PRODOM, VFAM. And tools in sequence assembly and annotation. Okay, that I haven't seen before. Let me check. Okay, only we have few left experiments. Like no, it will go for 2, 3 exercises. 2, 3 classes it will take, I guess. In next class we'll see SciBlast, FiBlast, CanProcyte, and PRAT. Together we'll see like generation of sequence profiles and derivation of sequence searching patterns. Like that one we will see. And in the following class, you will see the motif and domain analysis using MEMAST and eMotif, InterpoScan, ProSite, PODOM, and PayFab. Those things you will see. But I don't know about these tools in sequence assembly and annotation. Let me check it with sir. I don't know what to take in that one. Those things we can see up to 7, and we can see. Let's see what we can see up to like other things. Is it clear up to now? Like whatever I have taught you?
Yes, ma'am.
Yes,
Yes,
ma'am.
Yes,
ma'am.
ma'am.
Because in exam, you might, if you are here, like you'll be coming back to department, right? During December,
Yes, ma'am. Next month.
it will
Yes,
be
ma'am.
offline exam. So you will be here in department and you will write uh, in department. At the December, you'll be having your exams, right? At the end, I guess, maybe December end, you will be having your practicals or
Yes,
something.
ma'am.
So you will be facing Viva. Everything will be there. Uh, you will see all the faculties and like Viva will happen now. So you can manage, I guess. No problem in that one. That's why I'm asking you.
It
All the
is going
experiments
to
are well,
it,
tell.
it is going to difficult for us, ma'am. To be
Oh,
honest,
for the first time you're coming to department now, so
ma'am,
it
we
will be like that.
Ma'am, we are attending online class just to take the notes. We do not have much resources to read all the.
Yeah, I understood because like for doing the simulation and all, it's very difficult for you to do in your laptops.
Yes, ma'am. We haven't. We do not have much computation or in our laptop.
Computation and network facility won't be possibly like outside is very like difficult for network possibility. Here in department we are having good facility, so that's not a big problem. But in outside it's a bit difficult to learn it
Well,
from the
it
laptop.
is.
It's very tough, and you people miss the campus. That's what I say. So you'll be coming back now, so no worries. You can enjoy the rest of the times here. Okay, try to like brush up those things. Whatever I have taught you. Uh, I think you'll all be able to perform. But do you know you should be prepared for the questions also. It won't be a direct question. Perform this thing, perform those things. That won't be there. Okay, question pattern won't be like that. They will ask you how you have to identify the database for what we have to use, like for what. Okay, remember that also. Like while teaching the things, like it's fine. You know, like oh, we are doing ORF and we are doing emboss and we can tell them. But any tool you can use, that is different. But the questions won't be direct. Perform, Caspi, they won't say like that. Okay, the question will be like that only. Already, have you done any practical exams in online? Uh,
Yes, ma'am.
then you might know, right? Like how the question paper will look for our department.
Programming part we have done, ma'am.
Programming part only you have done.
Yes, ma'am. All other labs are like we are in this sem only we are doing the pending
Okay,
labs also.
structural biology also doing by online.
Structural biology we
That
did
was
already.
cancelled, ma'am.
Yes, ma'am. Internal we gave. Actually, sir asked Viva and everything, but the exam it was cancelled actually.
Oh, without okay because that tool you can't access now that WinGX software. Is it because of that
No,
one?
ma'am. At
Hmm.
that
All
time,
exams were
the
cancelled.
UGC told to cancel the exam.
Oh, again you have to write or you don't need to write?
No, ma'am. It
No,
was
no,
scored according
no,
to the
no.
internal marks. It was
Oh,
done
okay,
actually.
okay. Done this it's fine. No, no, yeah. Pattern will be different. For you people, like sexual biology paper will be different. For any people, sexual biology paper will be different. The practical session will be different. They didn't taught you about that WinGX tool, right?
No, ma'am.
Okay, then that will be different because now that tool you can't access by your system. The installation and all, it's then only in department. That's why I asked you like whether you did sexual biology or not. No problem. That according to syllabus it will vary. Okay, anyway, we will come back right by the time we will. I'll meet you people. Okay, yeah. Today class we can wind up.
Yes, ma'am.
Okay, ma'am.
Okay. For this only like girls are saying yes, ma'am. For other questions they are not opening their mouth. It's fine. Okay, try to finish all your experiments, and in next class, don't say that I didn't finish or like, especially Satish. Don't say that you didn't finish, ma'am. I'm
Okay, ma'am.
doing like that. You have to
No,
show
no,
your
I'll
results.
complete. Yes, ma'am. Yes,
Yes.
ma'am.
And whoever didn't finish. Oh, what about Chandani? Like, did you able to finish the other things? Okay, try to complete in this week. Talk with your classmates. Yeah, talk with your classmates and what are the things they have. Like I have given, you just get the IDs from them and try to finish it. Yes, everybody try to finish it soon. It's anybody absent today? Like any other people like miss the class?
Yes, ma'am. Actually, Madangi she is not feeling well. Actually,
Okay,
she
only
told
one
me.
person.
And
To-
Udayavan and Vishal, I didn't
okay,
see their names here.
okay, okay. Totally like 18 people, right?
Ma'am, Vishal is in hospital with his
Okay, no
mother.
problem. If that is their problem, that's up
Yes.
to them. Like I'm just asking, total number of like strength is 18.
No, ma'am. Actually, it is 17, but for this course we are 16 actually.
Oh, why? So,
One person is actually our senior. He didn't complete his second year, so he's
okay, okay.
doing that course alone. He already did this course offline mode.
Who is that?
Subhash is his name is Subhash.
Subhash is. Okay, I don't know. Okay, let me see. Check it. He is not attending the class. Oh, he finished this paper.
No, he already finished. Yes, ma'am.
Okay, okay, he
Yeah.
finished this
This
paper.
is during his first year, right? We
Yeah,
didn't
yeah.
do that part, so we are taking this course now.
Okay, okay, no problem. Maybe I haven't seen you as seniors. I don't know about that. Okay, it's fine. You just guys carry on and try to finish your works. Okay. Bye.
Sure,
Okay.
ma'am. Sure, ma'am. Thank
Okay.
you.
Thank you.